I've always wondered, why don't humans have a tail? Why do some of our primate cousins have them, but others don't? And what is the underlying genetic element that causes this? The popular science article, Tail Loss in Humans, Apes, Linked to Transposable Element Impacting Splicing, summarizes a study that analyzed the genomes of many primates with and without tails to understand what happened on a genome level and why exactly it was evolutionarily advantageous to ditch the tail. First, it's important to understand the basics of primates. Hominoids are primates without tails, such as gibbons, gorillas, and humans, while non-hominoids are comprised of Old and New World monkeys, as well as animals like lemurs. The study highlighted the use of analysis of protein-coding orthologs of primates via tens of thousands of SNPs to try and find a common genetic trend within the two groups that may indicate what caused the loss of the tail on the genome level. However, they came up empty-handed. It was in investigating the non-coding elements, however, that they discovered a common link, the ALUY transposon, which was inserted into a gene relating to tail development called TBXT roughly 25 million years ago, aligning with the split between hominoids and non-hominoids. Not only did this inform me of the importance of ALU elements in gene regulation, but also challenged my belief that most differences in gene expression are due to changes in protein coding regions, which, although already covered in GEN2052, it gives me a tangible example as to exactly how gene expression can be altered without changing that exonic sequence. Further, I challenged my understanding of transposons. I generally just viewed them as this weird quirk of nature that didn't really serve much of a purpose, but as I discovered, it is paramount to human evolution as we know it. In fact, this short interspersed nuclear element transposon, ALUY, acts by forming a stem loop with an upstream ALU element, which enables an alternative splicing event to occur, moving an exon outside of the transcribed DNA sequence. This is why the researchers couldn't see any consistent differences in the exon sequences between primates, because there isn't any. The study also uses mouse models to understand how altering TBXT genes impacted their development. The mice were given both full-length and exon-skipped isoforms of the gene, orthologous to what is seen in hominoids. The impact was that they either had small tails or lacked them entirely. There are limitations in using animal models, as it may not function exactly the same in humans, but it can paint a general picture of what may be happening. However, it is important to note that in this particular case, they didn't actually implement the transposon into the mice, just replicating its action, which can introduce inconsistencies. Further, only a single gene was analysed when many genes contribute to the formation of tails, so it's difficult to draw definitive conclusions from this study alone. Intriguingly, it was discovered that many mice with TBXT genes orthologous to humans also displayed neural tube defects. This could indicate that, despite the loss of a tail now possibly leading to genetic defect 0.1% of the time in humans, that was a worthy price to pay for bipedalism and the evolution of hominoids as a whole. Excitingly, this discovery opens up many research pathways to discover the root cause of conditions such as spina bifida, and may offer solutions for gene therapy for these conditions in humans and animals alike. More broadly, the article has shown me the possibilities in exploring non-coding regions in relation to genetic disorders and evolutionary changes over time, expanding my understanding of the techniques available to analyse the genome as a whole. In summary, the lack of tails in hominoids, as caused by the insertion of the signed transposon ALUY, is an example of transposons altering the expression of genes, specifically through ALU elements. The research to discover this highlighted the importance of not only exploring protein-coding genes, but also non-coding regions to determine similarities between species. Finally, it highlighted the use of mouse models to detect gene function, opening up research possibilities surrounding not only applications of non-coding regions for analysing gene function, but also in the connection between the TBXT gene and the occurrence of neural tube defects.
Go back! I want to be monkey!